Hello students, nice to see you people again. Okay, today we are going to study the ultrastructure. We have already studied the general structure of the muscle fiber, the general appearance under the light microscope. Then we have seen the high magnification of the muscle fiber, and we know that the muscle fiber is made up of different types of proteins, which are known as actin and myosin. Today we're going to see the basic arrangement, how these myosin and actin they are arranged. We're going to see the arrangement of the myofilaments. We have two types of myofilaments, which are known as actin and the myosin filaments. We know that the muscle fiber is made up of muscle fibrils, and the muscle fibril is in turn, it contains the myofilaments, which are of two types. One is the thicker myofilament, which is known as the A band, and it appears darker in color. And then the thinner myofilaments, which is known as the I band, and it is lighter in color. This is anisotropic and this is isotropic. The A band, which is a thick filament, is 16 nanometer in diameter, and we see that the myosin protein is present in the thick filament. The myosin protein is actually made up of two polypeptide chains, which are coiled together, and the tails are directed inward, and two globular heads, which are known as the cross bridges, are sticking outwards. These globular heads, the function of these globular heads in the thick filament is to link both the bands during the process of contraction when the muscle is contracting. The thin filament is 7 to 8 nanometer in diameter. It is made up of actin protein, and it is actually two strands which are wrapped around each other. And on top of these two strands is present another protein which is known as tropomyosin. Tropomyosin is another band of protein, another ribbon of protein which is wrapped around the actin molecules. These actin molecules are actually known as the G-actin. They are known as the G-actin. And these actin, when they attach around each other, then they form a fibrous filament. And this fibrous filament is called the F-actin. Around the F-actin, then we have the tropomyosin, which is wrapped around. And this tropomyosin, on top of the tropomyosin, then we have a troponin, which is a 3-protein complex that attaches on top of it at different areas to hold it in place, to hold the actin in place. Each myosin filament is surrounded by 6 actin filaments. Each thick filament is surrounded by 6 thin filaments. The thick filament is made up of myosin, and the thin filament is made up of actin, G-actin, converted into F-actin when they roll around each other, and then one band of tropomyosin is rolling around the F-actin, and then we have at various area positions, we have the troponin, which is a 3-protein complex. Now, look at this. This is the arrangement of both of the bands. Both the thin and the thick filaments are arranged in the sarcomere to give us a light and the dark band. Now, this is the dark band, which is the A band, very clear, and this is the I band. The dark band is known as the A band. It is anisotropic. It consists of myosin filaments. Now, these are the myosin heads which are sticking out. I have just told you people. This is the area where the actin is overlapping. So it appears to be a little bit thick, darker. And this is the area where the actin is not present, where we just have purely the myosin filaments. This is the Z zone, the H zone, sorry, it is called the H zone. And in the center of the H zone, we have the M line or the bare zone. It is known as the bare zone. It has a lighter strip in the middle, which is called the H zone. The heel, which is for light, it is lighter. And it is bisected by an M line, which is known as a median line. This is the heel zone, the median line. This is the A band. And the A band has the actin filaments around it. Now, this is the I band. In the center of the I band, we have the Zuyshen, line of Zuyshen, the Z line. The distance between one line of Zuyshen and another line of Zuyshen, the next line of Zuyshen, is called the sarcomere. And the sarcomere is the contractile unit of the muscle. The light band, which is I band, is isotropic. and it consists of actin filaments. It has a midline, which is called the Zushane, line of Zushane, which means it is present in between the discs. Structure, each sarcomere is made up of two types of myofilaments, the actin and the myosin. The myosin filaments are thick, 16 nanometer in diameter, and made up of a protein which is called myosin. Now, myosin molecule, it has two polypeptide tails, look here, which are rolled around each other, and they are pointed towards the center, and it has two globular heads sticking out. The myosin tails are arranged to point towards the center of the sarcomere. They do not stick out, the tails, and the heads, they stick out. These heads, they serve to create a connection with the actin during the contraction of the skeletal muscle. The tail has two long polypeptide chains. The heads are also called the cross bridges. We're talking about myosin, which link the thin and the thick filament during the muscle contraction phase. Now, the actin filament, look here now. This is the G actin, which is rolling around, and it is forming the F actin. This G actin, again, it is surrounded by tropomyosin. And at various places on the tropomyosin, we have a 3 protein complex, which is called the troponin. Troponin is made up of 3 types of molecules. And these 3 molecules, they have different functions. Actin molecule, 7 to 8 nanometer thick, chiefly composed of the protein actin arranged in 2 twisted chains. Twisted around the actin are 2 chains of a 3 polypeptide complex, which is the protein tropomyosin. Tropomyosin is twisting around the F actin. Then next, a third protein, the troponin, which is a 3 polypeptide complex, is present on the tropomyosin at various areas. The troponin has 3 types of protein complexes. One is TNI. TNI is actually inhibitory in nature. It is inhibitory in nature and it binds to the actin. It binds to the actin. This one is TN1. Then the second one is TNT, which binds to the tropomyosin, TNT, which is binding to the tropomyosin. And the third one helps to position it on top of the actin. And the third one is TNC, which binds to calcium during the process of contraction. When you study muscle contraction, you will understand how it is functioning. Each myosin is surrounded by 6 actin filaments. Sliding filament model is a topic that we will be completing next time. Till now, this is all for the lecture. Let me go to the main screen, and then from there, we can close the class. If there's any question, do drop the question in the comment box below.